In the vast universe of science, few stories are as fascinating and controversial as that of Rosalind Franklin, a British chemist whose work was crucial to one of the most important discoveries of the 20th century, the structure of DNA. Born on July 25, 1920 in London, Rosalind Elsie Franklin demonstrated from a young age a brilliant mind and an unshakable passion for science. Her path led her to study chemistry at Newnham College, Cambridge, where she graduated in 1941. However, it was her subsequent work in X-ray crystallography that would catapult her into scientific history, though not in the way she might have expected. In 1951, Franklin joined King's College London as a research associate. There, she embarked on a project that would change the course of molecular biology, the study of DNA structure. With exceptional precision and skill, Franklin perfected X-ray diffraction techniques to obtain clear images of the DNA molecule. Franklin's work was methodical and rigorous. She designed and assembled a special microcamera to capture high-quality images of DNA. Her dedication paid off when, in May 1952, with the help of her student Raymond Gosling, she managed to capture the famous Photo 51. This image, the product of 100 hours of exposure under carefully controlled conditions, clearly revealed the helical structure of DNA. Franklin made crucial discoveries. She determined that DNA could exist in two forms, A and B, depending on its water content. Photo 51 captured the B form of DNA, which would turn out to be the key to deciphering its structure. Additionally, Franklin concluded that DNA had a helical structure with multiple strands and phosphates on the outside, an observation she shared in a seminar in November 1951. However, the story took an unexpected turn. Without Franklin's knowledge or consent, her data, including the crucial Photo 51, were shared with James Watson and Francis Crick, who were working on the same problem at the University of Cambridge. This information turned out to be the final piece of the puzzle that Watson and Crick needed to propose their famous double helix model of DNA. In 1953, Watson and Crick published their model in Nature, along with an article by Franklin supporting their findings. However, Franklin's contribution was not fully recognized at the time. The story became even more complicated when, in 1962, Watson, Crick, and Maurice Wilkins, Franklin's colleague at King's College, received the Nobel Prize in Physiology or Medicine for the discovery of the DNA structure. Franklin could not be considered for the Nobel Prize, as she had died of ovarian cancer in 1958, at the early age of 37. Nobel rules do not allow posthumous nominations, which left Franklin out of this recognition. Despite this historical injustice, Franklin's legacy goes far beyond DNA. After leaving King's College, she continued her brilliant career at Birkbeck College, where she did pioneering work on the structure of the tobacco mosaic virus. Her contributions to understanding virus structures were fundamental and widely recognized during her lifetime. Rosalind Franklin's story is a testament to perseverance, scientific rigor, and passion for discovery. Her work was not only crucial to understanding the structure of DNA, but also laid the foundations for advances in virology and molecular biology. In the decades following her death, recognition of Franklin's contribution has grown significantly. Today, she is celebrated as a pioneer in her field and a symbol for women in science. Her story reminds us of the importance of recognizing and valuing the contributions of all scientists, regardless of their gender or circumstances. Rosalind Franklin's life teaches us that scientific progress is often the result of collaboration and the cumulative work of many dedicated researchers. It reminds us that behind every great discovery are countless hours of meticulous work.